it's a pleasure to be here presenting and also listening to all these excellent presentations. I do think that a vaccine will be the best solution for controlling this pandemic. I think we will live with this disease for a very, very long time, and the way to come back to the way life used to be in 2019, you need to have a vaccine. And there are a lot of vaccines being developed, probably more than 100 different vaccines that are being tried and are currently in different stages of preclinical and clinical development. Some of them shows like early signals of immunogenicity that are very encouraging in using very new technologies. So many different types of vaccine formulations have been tried, like from inactivated virus, viral-like particles, recombinant protein, genetic vaccines, either with DNA or mRNA. That's one of the most advanced examples is an mRNA vaccine. Also nanoparticles and some tableted heat inactivated virus that you basically eat for enteric release of the antigens. And you also have live attenuated vaccines using viral vectors like lentivirus, adenovirus. There is also bacterial vectors like bifidobacterium longum, and a variety of dendritic cell-based vaccines. There are some other vaccines that have been tried that are not exactly or specific for coronavirus. That is the BCG that may have an impact on boosting the overall innate immune response. There is also the passive immunization, like we just heard, the convalescent plasma, monoclonal antibodies, and also T-cell therapies that are being tried. There is already about 20 vaccines entering phase 1 and 31 that are already in a mixed phase 1-2 trial combined. And these are enormous number of vaccines, and the speed that has been developed from prototype to first in human has been extremely reduced. And normal vaccine development, or at least in our more recent experience, has been taking around 20 years or almost 20 years. And one clear example that I can share the experience is the development of the tetravalent dengue vaccine by Sanofi, Dengvaxia. And you started with the discovery of the yellow fever dengue chimeric virus. And from that went to the animal testing, and there are a very large number of different animal tests, which were very successful. And several aspects of the immunogenicity of the vaccine and protection levels in animal challenge and all that were successful as well. And then comes one critical part of vaccine development that is not only in regard to the biodistribution, toxicology, teratogenesis, all these things, but also the assays to measure the efficacy of the vaccine and to measure the safety of the vaccines. And during this development, in the example of dengue, neutralization or neutralizing antibody titers were the preferred method. and they shows standard generic cells, viral cells, which is able to demonstrate that the serum can block the virus to entry those types of cells. And at that time, there was a strong belief if you have neutralizing antibodies, you are protected. So the phase 1 trials show great immunogenicity, showed uh, a good safety profile, no significant adverse events. On phase 2, they started with, based on the encouraging results of the phase 1, they start immunizing significantly large numbers, including children. And the phase 2 trial was also encouraging to some degree, but also showed that the vaccine was only partially effective. Although that was not sufficient to stop the use of the vaccine, they started a phase 3 trial and combined multiple phase 3 trials and phase 2 trials. In total, they tested the vaccine in about 40,000 individuals. And 2 years later, or 3 years later, they were with sufficient data to show protection and essentially no risk of the vaccine. And the vaccine was licensed in a few countries, and they started rolling out. There was some restrictions or limitations in how the vaccine would be released, and the context was because they saw more efficacy in individuals 9 years older, or 9 years or older. They recommended vaccination only in patients of this age. And then by the time the vaccine was registered and distributed, things changed. And then the normal aspect of it, especially with new vaccines, you have a very strict follow-up guidance on how to use and monitoring specific problems, and they also continue doing some phase 4 studies. And that's a point that I want to highlight. Here you see a graph, and you see 4 lines. 2 are dotted lines. The dotted lines are from individuals that received the vaccine, and the continuous lines are individuals that receive placebo. And in this case, the red lines are individuals that were seropositive for dengue at the time of immunization. What it means is that they had dengue at least once before they took the vaccine. And in the other group are individuals that never have been exposed to dengue before, that are the blue lines, and took the vaccines. They were 9 years of age or older, and they never had dengue before, and they took the vaccine. So if you look into this graph, you're gonna see the immunized individuals versus the placebo, and the red are the people that had the primary dengue infection and took the vaccine. Had a huge benefit in this group, more than they had quantified previously. But when you look at the individuals that were naive for dengue and took the vaccine, you see very modest effect up to 1.5 years after the vaccination, and then things start to change, And then you start to see that immunized individuals have now the greatest risk to develop dengue hemorrhagic fever or severe dengue, much more than the individuals that had no vaccination. So in this, I would say it's good and bad news. The good news is that if you had dengue and took the vaccine, you were protected. Uh, However, if you had never had dengue and took the vaccine, you had greater risk. And what does it mean is that after this period, after this date, the way the vaccine is recommended to use requires previous testing of the individual, if they have been exposed or not. And some difficulties there are some difficulties in this testing because cross-reaction with other flavivirus, you require an assay that is very specific for dengue and doesn't cross-react with others. And there are a few major points, or 5 major points I want to bring attention to, is that in vitro neutralizing titers show a limited correlation with protection. In this Sanofi vaccine and other vaccines like the one by Takeda, it shows that even if almost 100% of the individuals develop neutralizing antibody titers against all 4 serotypes. The problem was that the levels induced or the lack of proper T-cell responses was not able to provide protection. You see also a limited duration. You see that even the titers decay over time. The titers of antibodies induced by the vaccine decay over time quite significantly. And the efficacy was highly dependent if there was previous exposure or not. So, and the behavior observed in the immunized group that was naive to dengue, it supports the antibody enhancement theory that I will explain more later. The good aspect of it is that you can use the vaccine if you do proper screening. You could use the vaccine safely and with good results. But the point I want to stress here is that take some time to fully observe the effects of a vaccine. It's not immunizing today and 2 weeks or 3 weeks from now doing some challenge or measuring efficacy after immediately after immunization. You have a lot of in a specific response or innate immune response there that has an impact on the clinical outcome, and it's not truly dependent on the memory that the vaccine has induced. So this graph in the top line is the antibody titers of individuals that took the vaccine 5 years later. The titers are not that impressive. When you look in the placebo group that was seropositive in the baseline, the difference is very little. It's not that big. And when you look to the the neutralizing titers of individuals that that were seronegative and took the vaccine are much lower than the ones that were seropositive. And if you look at the ones that took placebo, the titers are very, very low or almost neglectable. But these levels were a little bit higher and apparently more capable to induce ADE. So What is antibody-dependent enhancement? What happens is that you have antibodies that can bind the virus, but they are not sufficient to neutralize or kill the virus or opsonize in a way that destroy the virus. So these immune complexes can be captured by immune cells that express the Fc gamma receptor. There is a receptor that binds specifically to the immune complexes. And when this is internalized in a certain way, it ends up increasing the infection of immune cells, and also has an impact on the antiviral responses of the cell. So the antiviral response of that particular cell are reduced, which facilitates the viral replication, and those cells end up being able to produce more virus. Now, coming to the COVID-19 vaccine development, everything is being done in parallel. The prototype vaccine to animal testing clinical trial is done almost simultaneously. So you make your chimera, your plasmid or your mRNA or your DNA, and you start animal testing and developing the assays to measure the immunogenicity of the vaccine in the phase 1-2 trials simultaneously. What it means is that you don't take the experience from one phase to the next. You are learning at the same time everything. So, in one key aspect here, it's because at this stage, the main parameter you have to judge if the vaccine is being good is actually the assays you use to measure that neutralization, the T cell responses, and they need to be validated properly. And this takes time. You require gold standard, gold-plated reference samples that require excellent characterization and all that. And it takes a time. It's not that these panels are really available. So one way that has also been proposed to speed up the tests is to conduct these structured human exposures, but translating to regular colloquial language is a human challenge. Basically, what you do is to immunize individuals, and then you expose them to the COVID-19 virus and study their outcome. Basically, what is happening is taking individuals from the phase 1-2 trials and selecting a subset of them to do the challenge. And the expectation is that if successful in these individuals, the human challenge, which may happen by next spring, we'll start rolling out the vaccine. And you're gonna need to be very careful at this stage, because in essence, it's still almost like a phase 3 trial, a regular phase 3 trial. And you may start with very, very high risk individuals, like people with 95 years of age or older, with comorbidities, and perhaps healthy healthcare workers that are highly exposed. And you note that until this stage, you not have even given the proper time to evaluate how long the immune response lasts. And also, there is very little understanding of the immune pathogenesis of the COVID-19 at this stage. So, and after that is that you start licensing and expanding the use and for general population, which may be essential to actually control the outbreak. So at this stage, the correlates of immunity or the mechanism of how the immune system can protect you against infection is still limited, the understanding of this, and much less what is causing the disease. Like we had very clear examples mentioned in the previous presentations about the microthrombos, these cytokine storms that are involved in the pathogenesis of the disease. So at this stage, it's very important to identify the epitopes and that are correlated of immunity, to identify the antibody subclasses and kinetics of the virus. So another aspect that for COVID-19, mucosal immunity may be essential. And we need to study this profoundly. We still don't know. And also to investigate the possibility that antibody-mediated enhancement it could be occurring in COVID-19. We also need to study the role of T-cell responses. The same, identify T-cell epitopes, characterize the types of CD4 T helper responses and CD8 responses. So the mechanisms associated with pathogenesis need to be better understood in terms of cytokine storm or complement activation. What is causing that? That have reported antibody-dependent enhancement against coronavirus to SARS-1 and to feline coronavirus. They have been shown to cause in these animals peritonitis. Monoclonal antibodies have been shown to cause antibody-dependent enhancement. Has been shown to occur also in macrophage, in alveolar macrophages and human monocytes. In the case of coronaviruses, has a different aspect than in dengue. In the coronavirus, the antibody-dependent enhancement has also been demonstrated in homotypic fashion. What it means is that you have immunization or infection by one antibody, by one virus, and you end up having a worse outcome after reinfection by the same virus, which is different than dengue because you change serotypes. So the mechanism here may be slightly different. And the same or similar mechanism has been shown for SARS-1 as well. So how it works in COVID-19. So you have this, the virus particle, and you have antibodies that can completely block the spike protein from binding to the ACE receptor and entering the cell. However, there are other possibilities that antibodies that do not neutralize the virus and form these immune complexes and actually generate the immune complexes just like the dengue example, and that can be captured by the Fc gamma receptor and facilitating the infection of those monocytes and macrophages. There are other types of mechanisms that can happen as well. Once the infection and the virus is incorporated in a multimeric fashion, those cells or dendritic cells, they, in addition to facilitate infection, it also inhibits the innate immune response, the antiviral response, and also you start to produce this enormous amount of pro-inflammatory cytokines, generating an imbalanced response. And here is an example where naive serum is presented, is administered at different concentrations, and you see a constant rate of infection. When you use an anti-spike protein serum, you can see protection at high concentrations that diminishes as the tighter the amount of antibody diminishes, reduced. And by once the concentrations get low enough, you actually have several fold greater number of cells infected. There are two factors that interferes with the, let's say, the interactions on how they occur. In the case of heterotypic antibodies like dengue 1, dengue 2, there is the effect of the affinity, because the antibodies against one virus don't necessarily are identical to the other virus, but they are similar and they can bind to that with a lower affinity. So concentration and affinity makes a very big play an important effect in ADE like flavivirus. In the coronavirus, you can have, I would say, the two situations as well. But what has been demonstrated so far is the homotypic, that is mainly dependent on concentration of antibodies and not necessarily on the affinity of that. And however, we cannot rule out the heterotypic ADE, because although the similarities between the regular human coronaviruses and SARS-2 are small or not necessarily, but are not neglectable, the homology is in the range of 50%. Although the homology is not that great, if you have very, very high concentration of antibodies against endemic virus, you could have similar effects of perhaps some degree of protection or perhaps some degree of enhancement. And here, based on different situations, as I point out here, if you have high affinity or high concentration of antibodies, you can completely block the infection. The worst case scenario was when the proportion of antibodies and virus are equivalent, and you can form networks of virus that have not been neutralized, but can be easily taken up by cells that infect, by cells that express Fc gamma receptor. And the other situation is that you have such low affinity and low concentration that the presence of those antibodies are indifferent. So we have to pay attention to that. Here is the profile of the immune responses to natural infections. Here, it's relatively small. We have very few studies at this point showing the kinetics of the antibody responses. In this case, for IgM, these are all PCR confirmed cases. You have the majority of individuals didn't produce IgM. fraction of them produce a very low levels of IgM, and I would say less than 30% produce high levels of IgM. I would say the fact that many individuals never elicit IgM response, to me, it's a bit concerning for two reasons. Why is that? It's because we are not having enough activation of naive B cells, or is it because what is happening is that we are boosting the memory B cell response to previous exposure to other coronavirus? We do see similar event in dengue. About 30% of the individuals in a secondary infection don't produce IgM. Only in the primary infection, I would say in contrast, in primary infection, almost 100% produce IgM, while in secondary infections, you have about 30% that never does. This is titer of IgG, and the titers here are not impressive. These are not neutralizing titers, and you see around 1 to 20 is the majority. This is borderline cutoff level, and you have some individuals that continue to be IgG negative, but the vast majority was a low level of neutralizing antibody in natural infection. And this we are counting only less than 2 months after infection. Only a smaller group actually produced high levels of antibodies. So in the context, in this context, I want just to remind that the closest is the antibody level in the serum to the dilution that leads to the peak enhancement. What it means is that, let's say, if I dilute you have a titer, IgG titer in your serum, let's say 1 to 40, and then let's say these titers drop in half, and by the time they drop in half, they started already enhancing. And this would be a very high risk situation as compared to a situation that you would have to, the antibody titers would have to drop, let's say 100-fold to actually reach peak enhancement. So how you can at this time reduce or at least start investigating the risks involved in the COVID-19 vaccines. My first suggestion is also based on the dengue experience. One of the factors that was afterwards considered that was misleading during the vaccine development was the use of viral cells as the standard to measure neutralization. It's essential to use more biologically relevant cell types. In the case is using human, lung, or epithelial cells, or any kind of pneumocytes. And the viral cells are kidney cells that they are used because, or the viral E6 is used mainly because express high levels of ACE2 receptor. But it's not the primary cell target that is involved in the infection. So cells from the respiratory system, either mucosal or lung, are essential to have that. And especially cells that express different types of Fc gamma receptors or FcRn, that it's another type of receptor that is involved in transport, in transcytosis in the placenta, but it's also present in the lung. Another aspect is that the human challenge studies should not be necessarily restricted to the individuals that developed the highest immune response or at the peak of the vaccine response. It should be also conducted in later time points as the vaccine, the immune response decays, or also test the challenge in individuals that received a low dose of the vaccine or produce low levels of antibodies in response to the vaccine. This is a complicated ethical question. However, would be also unethical to release a vaccine to the wide population without knowing that it may be 6 months or a year later, you could actually be setting up those people to a more severe form of the disease. So currently, I have been focusing my research in trying to understand the mechanisms of immune pathogenesis of COVID-19, especially the role of antibodies against the human coronavirus in comparing the early immune response to COVID-19 and other coronavirus to the clinical outcomes, and also studying the antibody-dependent enhancement that this serum can cause, and also characterize the complement activation at early stages. So with this, I hope to be able to develop ways to access the safety or potential or identify risks, or at least to identify ways that I can use to evaluate if the vaccine is efficacious and safe to use. We're gonna need those tools down the road once the rollout will start. So with that, I just come to a few conclusions that there is an urgent need to study COVID-19 correlates of immunity and pathogenesis. We need to develop assays that are equally capable to measure neutralization and antibody-mediated enhancement in ex vivo and animal models. And also we need to carefully design human challenge studies that can address the impact of low antibody responses that will occur after a few months or a year that the vaccine has been administered.